The last macromolecule is nucleic acid. This is the macromolecule that makes up our genetic information. It comes in two broad forms, DNA and RNA, and as we'll see in just a minute, the differences are just with the sugars that are present and then bases. You remember, hopefully, gene expression from the first chapter where DNA is converted to RNA by that process known as transcription, and then RNA provides the instructions for making protein by the process known as translation. So here's the DNA molecule as a double-stranded molecule in the nucleus of this eukaryotic cell. RNA is a temporary copy of that DNA. The RNA molecule exits the nucleus and moves into the cytoplasm where it is translated. The site of translation is a ribosome, that organelle on the cytoplasm where proteins are made. So the sequence of the RNA directs the synthesis of the protein by directing which amino acid goes first, which amino acid goes second, and so on like that. So that process of translation and production of a protein happens on a ribosome. So to look at the polymer structure of, well, polymer and monomer structure of a nucleic acid, let's look here. So on the left here, we have the polynucleotide or polymer form, which is made up of repeats of this monomer form, which is called a nucleotide. And nucleotides are made up of three components. A phosphate group you should recognize, a sugar, it happens to be a 5-carbon sugar, and then a base with some nitrogen associated with it. This is called a nitrogenous base. And I'll come back and show you this again in just a few minutes. The nucleotides are joined together end to end in a linkage we call a phosphodiester linkage. So there are two ester linkages with a phosphate in between. And so the sugar backbone connected to the yellow here is the phosphate group, then connects to the next sugar, and then there's a phosphate group in between, connects to the next sugar, and there's a phosphate group, and so on. So a strand of DNA or RNA has a distinct beginning end, that's a phosphate, that's attached to the 5' carbon of this sugar, so that's called the 5' end, and then you have the 3' carbon of the sugar attached to the next phosphate group, to the 5' carbon, to the 3' carbon, to the phosphate, and so on. And at the very end, where there's no sugar attached, is the 3' hydroxyl. So all nucleic acid chains have a 5' phosphate on one end, and so we just call this the 5' end, and a 3' hydroxyl on the other end, and we call that the 3' end. So they have a distinct beginning and a distinct end. And the backbone is the sugar-phosphate, sugar-phosphate, sugar-phosphate linkage. The nucleic acids face into the center, and we'll see those in just, the nitrogenous bases in the center. So here are the variations on the nitrogenous base. So this is what gives our DNA or RNA those letters, G, A, T, C, or U, is the type of nitrogenous base that's present. And so we have these two double ring structures known as purines, adenine and guanine, and these three single ring structures known as pyrimidines, cytosine, thymine, and uracil. And so we use the letter abbreviations to represent those nitrogenous bases. And so in a chain of DNA or RNA, you have a sequence of, this could be A, C, T, C, for instance, a sequence of these nucleotides, and the composition of the nucleotides varies based on what nitrogenous base is present. Then the difference between, one difference between DNA and RNA is that DNA has thymine and RNA has uracil, so you don't have T's in RNA, but rather you have U's, and you don't have U in DNA, but rather you have T. And then the other difference is the type of sugar present. They both have a 5-carbon sugar, but if you look right here on this second carbon, in deoxyribose, there's just a hydrogen there, and in ribose, there's a hydroxide group there. So deoxyribose is missing that oxygen, hence the name deoxyribose. but otherwise the sugars are the same. So again, here's the structure we were just looking at with the distinct 5' and 3' end, with the phosphate sugar, phosphate sugar, phosphate sugar backbone, and then the purine nitrogenous base or pyrimidine nitrogenous base attached to the sugar. DNA is a double-stranded molecule, so the two strands are run parallel to one another, actually anti-parallel, because the 5' end of one strand is attached to the 3' end of the partner strand. And the base pairing rules are such that cytosine base pairs with guanine, adenine base pairs with thymine. And so you get the attachment between the double strands due to hydrogen bonds that form between those base pairs. RNA is a single-stranded molecule, so one end of this is going to be the 5' end, the other end is going to be the 3' end. It can fold back on itself and have those same kinds of hydrogen bonds, but it's just one strand of RNA total. So here's the anti-parallel nature of the double helix or the double-stranded DNA molecule. Here's the 5' end of one strand and the 3' end of the other. This strand is 3' on top and 5' on the bottom, so they run in opposite directions of one another. And then we see the base pairing rules here. There are 2 hydrogen bonds between each adenine and thymine, and 3 hydrogen bonds between guanine and cytosine. So that's what attaches these together. And you might notice here as well that each of the base pairs is one purine with one pyrimidine. So purine to pyrimidine pairing. So the information in DNA is really in the sequence, and the sequence of DNA, remember, directs the synthesis of RNAs, which then direct the synthesis of proteins. So DNA contains the information about how to generate that primary sequence of a protein, like I was showing you with the sickle cell, with the hemoglobin protein, for instance. So sequences in DNA direct the synthesis of proteins. And if you have a change in the DNA, like you would in that sickle cell example, that changes the protein that is produced. And so changes in DNA sequences are mutations, and mutations get passed down evolutionarily. And so you can use the frequency of mutations as sort of a measure of how much time has passed between 2 related organisms. So you're going to have a lot of DNA sequence similarity with your children and with your parents. But if you go back to your great-great-great-grandparents, there may be a mutation or 2 that has occurred between them. And then, of course, DNA from other individuals in that lineage are also going to contribute to your DNA. But if you want to compare, say, a dog and a cat, because they're evolutionarily related, you could trace back in time how distantly related they are based on how many mutations have occurred since they separated from one another. So DNA can really serve as a yardstick of evolution, if you will.